Hello and welcome to our next set of notes on biochemistry. In this set of notes, we're going to talk about nucleic acids and we're going to talk about proteins and how both are put together and what they do. We're going to start with nucleotides, which are the building blocks of your nucleic acids. Nucleic acids are DNA and RNA. Okay. So nucleotides build up your DNA and your RNA. Nucleotides are your monomer, and DNA or RNA are your polymers. So nucleic acids are the polymer, and their purpose is to store genetic information. There's two major categories of nucleic acids, DNA and RNA. DNA stands for deoxyribonucleic acid. That's what the DNA letters stand for. Whereas RNA stands for ribonucleic acid. Now these molecules we're going to talk about in a lot more detail later on in the year when we start talking about DNA structure, how DNA works, and what we use RNA for in terms of making proteins. The monomer for nucleic acids, like I mentioned earlier, is a nucleotide. Each nucleotide has three parts to it. A nucleotide has a phosphate group, which is a phosphorus and 4 oxygens. That phosphate group is attached to a 5-carbon sugar, and the 5-carbon sugar is attached to a nitrogen base. So the 5-carbon sugar is actually different for DNA and RNA. For DNA, your 5-carbon sugar is deoxyribose. Okay, so that's where we get our name from. And for RNA, it's ribose. Okay, so these are your sugars. The difference between these two molecules is simply that deoxyribose has one less oxygen than ribose does. The other thing that's different between your nucleotides is these nitrogen bases right here. There are, for both DNA and RNA, 4 versions of the nitrogen bases. There is, for DNA, we have A, T, C, and G. But for RNA, we have A, U, C, and G. The only difference in your nitrogen bases for DNA and RNA is this T versus U. Again, this is something we're going to get into more detail about later on in the year, so don't get too bogged down in this right now. But basically understand that monomers are nucleotides. The nucleotides have 3 parts, a phosphate, a sugar, and a nitrogen base. And there's 4 different monomers for DNA and for RNA. Okay, so we're going to spend the rest of the time talking about proteins. We talked about them a little bit in class already, but now we're going to talk about them in a little bit more detail. So proteins have carbon, hydrogen, oxygen, and nitrogen present in them. Their building block or monomer is what's called an amino acid. So an amino acid has 4 basic parts to it. One section of it is your amino group. So this area right here is your amino group. Okay. The amino group gives you the amine part of your amino acid. It's also basic. And so one side of your amino acid structure is a basic structure. This side over here is your carboxyl group. And the carboxyl group is an acid. Okay. So this is where the acid part of amino acid comes from. Your amino group and your carboxyl group rotate around a central carbon. Okay. So this is your central carbon. All amino acids will have attached to your central carbon will have a hydrogen atom. So the hydrogen atom is just a hydrogen. That's it. Nothing else. Okay. And then on the other end of your structure, you have this R group. R is a variable. So R can change in your 20 different amino acids. So for proteins, there's 20 different amino acids. What's different about those amino acids is this R group. The amino group, the hydrogen, the central carbon, and the carboxyl group are all going to be the same for 20 different amino acids. All that's different on these is the variable. So the R group is going to be different for each amino acid, and it's these differences in the R group that's going to cause a difference in the shape of the protein. Protein shape is going to determine protein function. That when we change the shape of the protein doesn't work anymore. So some functions of proteins, some of them are used for movement. So like we mentioned that your muscles are made out of protein, and the purpose of the muscles is to move the body around. Some of them are used for structure, meaning they're going to provide a support or a framework for things to grow on or to be built upon. Some of them are going to regulate, like insulin is a protein that controls and regulates your sugar level. And glucagon is another protein that's going to control and regulate your sugar levels. Some proteins are going to transport. For instance, hemoglobin is the protein in your red blood cells that transports oxygen around your body. Some are going to be used for nutrition. Your digestive enzymes are used to break down your food so that you can use your food to provide energy and resources for your body. And finally, some of them are used for defense. And this is like in your white blood cells, there's going to be special proteins in them that are going to be used to break down and destroy bacteria and viruses and other things that don't belong in your body. The problem for proteins is that they are very sensitive to both temperature and pH. So they will actually fall apart and become non-functional outside of a happy range for them, or what's called an optimal range. Optimal is a fancy word that means best. Okay? So for instance, proteins in the human body, they can't function over 106 degrees Fahrenheit. This is a bad temperature. In humans, if the brain gets up to 106 degrees Fahrenheit and stays there long enough, and it's not very long, I might add, then the proteins in the brain actually start to fall apart, and then the brain stops working, and it can kill a person. So that's why if your fever goes really, really high, they send you to the emergency room to get your temperature brought back down quickly so that there's not damage. So a dipeptide is formed when we link two amino acids together, and we hold them together using what's called a peptide bond. The peptide bond is the specific name for a covalent bond that's going to hold together two amino acids. So when I look at this structure here, I have amino acid over here, and then I have an amino acid over here. And what's holding these two amino acids together is the peptide bond in the middle. I can form another peptide bond on this side over here. I can take off the OH here and link up to another amino acid on that side. And I can form one on this side and have one going off that way. We get long chains of amino acids, which are called polypeptides. And these are formed when I put a whole bunch of peptide bonds together. Now, there's multiple levels of organization for polypeptides or for proteins. The first is the simplest, which is just the order of the amino acids. And then we start getting some funky shapes in here. We get what's called an alpha helix, which is a spiral. And you see that spiral here. So we get a curlicue, like curly hair. Or we get a beta sheet. Beta sheets look like your pleated skirts. From there, we get a three-dimensional shape. And then if our protein is made up of multiple polypeptides, I get a fourth level shape. We're going to talk about this in a lot more detail when we do protein synthesis, and we'll do some modeling activities. As always, if you have questions, please feel free to write them down and ask in class.